Please join me in welcoming Dr. Nadia Rosenthal. Welcome.
Thank you, Jennifer. Thank you and all your colleagues for this opportunity to share some of our exciting new research on COVID-19 and disease susceptibility. I'm going to start by taking you back to a time which you probably don't even want to remember, when we first heard about what was going on in the Wuhan district in China and realized that we were on the verge of a pandemic. And it was something very mysterious because it seemed to be a virus that nobody had ever seen before. And, I think we're all now aware of most of the facts that I'm going to start by telling you about, but I'm just going to review for you a little bit of what we know about the coronavirus pandemic that we are still in, and some of the attempts of scientists to address the problems that it has caused. This is a picture of SARS-CoV-2. It's enhanced. The little pink dots are the viruses attacking a cell. And that's sort of what happens when you get COVID-19. The actual virus itself is formed with nucleic acid on the inside, which is its genetic material. And then on the outside, it has a number of different protein structures. And the most important protein structure to understand what I'm going to tell you about tonight is this spike protein, which sort of looks like a golf tee. And there are many of them sticking out on the surface of the virus. And they are the point of attachment to human cells. So we're particularly interested in how this particular protein manages to invade our own cells. One thing that we do know is that there's a very broad disease spectrum. As we all have heard or experienced, we get infected in our lungs, heart and blood vessels can be involved, brain fog can develop, your eyes, your nose can be infected in the same way that a flu virus gets you, but also other organs can be attacked, such as liver, kidney, and intestine. So it appears that this virus has a very broad range of tissues that it can actually access. And this produces a very complicated patient spectrum of diseases. As we know, common symptoms include fever, loss of appetite, fatigue, loss of smell, very interesting, shortness of breath and cough, just as you would in a flu, and muscle aches and pains. But the severe disease is something that really is the problem that we face, which is we have no idea who is going to develop a severe disease. And it used to be that we assumed that all of these symptoms, difficulty in waking, confusion, coughing up blood, decreased white blood cells, and then often organ failure, is something that appears to happen in a subset of patients. So here's just a sort of an overview of what happens in a mild infection on the left and a severe infection on the right. So on the left, a mild infection, which is sort of what you get when you have a common cold, which is also a coronavirus. You end up with an immune response, inflammatory response in your lungs, and you feel sort of woozy. And that's because your immune system is kicking up and getting ready to destroy the invader. What happens in those of us who get severe disease on the right is that the immune system appears not to behave in the same way. It's a bit delayed, and then when it gets going, it overreacts. And that's a lot of what the sequelae of this very severe response involves, with the different tissues that you can possibly imagine being affected down at the bottom there. And the question that we're interested in is, why is there such a different response between people? As we know, the majority of us have not ended up with severe COVID. But for those of us who have, it's no mean feat to get over it. And of course, death is one of the issues that we're facing. In particular, at least in the beginning, with the increasing age, there appeared to be an increasing susceptibility for acute illness. And any chronic conditions also seemed to exacerbate the disease. So comorbidities such as diabetes or heart disease or any kind of autoimmune disease was considered a real warning sign. But even then, now we know that long-term effects, we call it long COVID, are unpredictable and are affecting people who got either mild disease or were young and didn't expect to have any kind of persistent effect. So this suggests that there is an enormous amount of variation in the way this same virus can affect so many different people. And we call this host response, the host being us and the virus being the invader. So the question that really became very acute for many of us in the field was, why is it that so many people have so many different responses? And is it the underlying genetics of these different groups of people that make some more susceptible to COVID-19? So about a year ago, these were some of the headlines in the newspaper. How sick will the coronavirus make you? The answer may be in your genes. Or could genetics explain why some COVID-19 patients fare so much worse than others? And then finally, the hopeful aspect of it, which is that people were beginning to pay attention to host genetics, to the genetics of the populations that were seemingly more susceptible to the virus, and trying to unravel what was going on with those people. And things like the COVID-19 Host Genetics Initiative was formed, a worldwide attempt to pull all the data together from as many patients as possible to try to find common clues as to why some of us recovered so easily and others did not. Now, all of this feverish work was, for the most part, done on humans. And the reason for that was it appeared to be a human disease. And we know that other organisms can actually get COVID-19. Of course, the famous bat story from the Wuhan live markets really persisted. But there are other susceptible animals like hamsters, and even some suggestion that domestic pets could get the disease, not as badly. So the question was, were there any models that we could use in the lab to study the differences between people who get the disease and don't really suffer from it and those who really get very sick? So I'm a mouse biologist, and the immediate question I had in my mind after that January revelation was, can I make a mouse model that will allow people to study the variability in COVID-19 responses? And of course, there's no question that mice are some of the premier organisms. First of all, they're mammals, so we share 98% of our genetic material with them. Secondly, they're very fast breeders, so that you can study the genetics by looking at heredity. And thirdly, they have been so well studied that we know tons about them already and are able to detect problems with their physiology and their disease responses very, very quickly and very completely. So it seemed to me that this was something that we could really use, especially given that we need a model to test new diagnostics. We certainly don't want to test them all on humans. And that's true also of therapeutics, vaccines, and antiviral approaches. So I'm going to tell you a little bit about how we approach this problem as a scientific community, and then a little bit of data from my own lab to tell you some exciting unpublished work about some progress we're making and how we think these mice can be extremely useful, and why it is that indeed we use mice in general for disease modeling. So you probably have heard all about this. Everybody has. It's amazing how much more we know about science as a community. People talk about PCR tests. Nobody was talking about PCR tests before the COVID pandemic. People are talking about making CRISPR mice, and they're talking about all sorts of other arcane sorts of phrases that I'm always astonished to hear coming from people that I pass on the street. but it's sad that it took this to get people interested in science. But the fact is that by now everybody probably knows that the way the virus enters your cells is through a receptor that you naturally make, and it's called the angiotensin converting enzyme 2 receptor. It's not really important what it's called, but just to know that it's actually a really important component of your own physiology. It actually breaks down another a gene product called angiotensin 2, which is kind of a hormone that controls blood pressure and blocks organ damage. So angiotensin 2 is actually the bad guy in this case. It increases your inflammation and it increases blood pressure. And so ACE2 is there to modulate the angiotensin 2 effects, which you need. You need control of blood pressure. You need to have your blood pressure higher or lower, and you need to have inflammation. But when angiotensin 2 goes nuts, you end up with high blood pressure. So the problem we have is that this virus is getting into the cell via the same receptor that normally responds to signals to tell it to go out there and control the angiotensin 2. So not only does the viral entry into your cells and the replication that follows depend on ACE2, but it also blocks the normal function of ACE2, which may explain some of the downstream problems we have in COVID-19. So here's just a couple of science pictures. This is what protein chemists do when they're trying to figure out how two different proteins interact. So remember I told you about the spike protein being very important, that little golf tee. That's the yellow sort of string, which is obviously a cartoon of the way the protein looks. And then the ACE2 protein, which is the receptor on the surface of the cells, is in blue. And you can see it has all sorts of fancy structure. But the important thing is where those pink arrows I've put in there. That's where the spike protein finds and interacts with the ACE2 protein. And by doing so, it changes the configuration of that protein, and the protein drags the spike protein into the cell with it. And along with that comes the virus. And that's how the virus gets in. Now, here is the problem we have with making a mouse model. Mice have an ACE2 protein, but it's oddly slightly different, even from a rat. So if you just look down that column, you don't really need to know what it all means. Those are just the names of amino acids in letter form. Amino acids are the building blocks of proteins. And the line there that has the numbers 31, 35, 38, etc., those are just the number of amino acids along the line from the beginning of the protein to the end, because it's a string of amino acids. And you can see that in the human, it goes K, E, D, M, K. And actually, there are a whole bunch of other interesting animal ACE2 sequences here. Bat, look at bat, the third one down, that's supposedly the animal that started the whole pandemic. And it actually has a few changes from the human, but somehow the ACE2 gene, the ACE2 protein of the bat is recognized by the SARS-CoV-2 spike protein. Now you look at mouse, and mouse has too many changes in too many key protein positions. The amino acids are different. So this is why mice don't get COVID-19. If you spray some of the virus up the mouse nose, it walks away sneezing and doesn't get sick. So this is a problem if you're trying to make the model. So one of the ways that scientists decided to approach this problem is to add the human ACE2 gene directly to the mouse. And we can do this with the kind of technology that now everybody's heard about, CRISPR and transgenics and recombinant DNA. Anyway, it's the sort of thing we do at the Jackson Laboratory on a regular basis. And this mouse here was actually made by a colleague who was studying SARS-CoV, not CoV-2, but 1. You probably remember the SARS epidemic a few years back. He made this mouse that had multiple copies of the human ACE2 gene, and it was driven by a very strong driver, a piece of DNA that turns the gene on in the epithelial lining of blood vessels and actually your skin as well, which is where ACE2 normally is expressed. Now, this mouse had lots and lots of human ACE2, walked around with it, didn't seem to mind. except for when it was infected with SARS-CoV-2. And then the mouse got really sick and very, very rapidly lost weight, which is one of the first things that happens in a mouse because they have to eat all the time. And if they don't feel well, they don't eat and they lose weight really quick. And it eventually would die very soon afterwards with respiratory failure. Well, you'd say, well, that's terrific. At least the mouse is getting sick. And that means if we give it a vaccine and it doesn't get sick when we infect it, the vaccine's good. And the same is true of any therapeutic like an antiviral. So it was great for those kinds of applications. But if you want to study why some people get sick and others don't, this mouse is not really doing it for the community. I'm going to show you a little bit of science data here, but don't worry, it's not very complicated. And furthermore, it's absolutely critical for you to see this and understand what it means so that I can explain how things go in the next couple of experiments I'm going to show you. So on the left, you're looking at something called a survival curve, and that's pretty easy to understand. So on the vertical axis, you see 1 to 100, and that means how many mice survived out of, let's say, you do 20 mice. And if 20 mice survive, then you get 100% survival, and the curve stays flat from 100, which is where you start. However, if the mouse succumbs to something, then you have a drop in the percent survival, like down to 40%. And that's the red line. And that's what happened when these researchers gave the mouse a high dose, 28,000 PFU. It's just a way of describing how many viruses they sprayed up the mouse's nose. And you can see that although dropping that number of viruses by tenfold, and that's the light blue line, it didn't really change the fact that the mice still died. So it didn't look like it was a dose thing. And as I said, the mice died very quickly. So in a week, they were all gone. And you can see it over on the left, how fast they lost weight. And if you're a mouse, you only sort of weigh about 36 grams. So if you lose even 20% of your weight and get down to 80% of your weight, you're already in bad shape. And of course, that's where those little dots ended, because that was when most of the mice were dead. So it's the survival curve on the left that's probably the easiest thing to read. And when I show you some of the data we got, you'll be able to very quickly pick up whether survival is good, and that means the lines are high, or survival is bad. So this is the way patients are also graphed out. Now, the problem with this whole approach was that we were trying to do something that was probably not possible, which was to find a mouse that would actually be appropriate model for all of us. And here we are, and we're all different, and we all have different responses. So the way that I think about it is, if you have one mouse and you're trying to model an entire human population, it's not a great sort of design. What you really need are as many mice. You need just as many mice that are as different as humans are from each other. And then you study the disease in all those mice. And if you have enough of these mice and you know enough about them, it's very simple to actually isolate the genetic basis of the problem you're seeing, because you can actually cross these mice and ask what their children have. And that's basically how we do genetics. So the concept is simple. Why look at one mouse when you can look at a whole bunch of them? Now, at Jackson Laboratory up here in Bar Harbor, Maine, where I'm talking to you from, we have 15,000 different mice, 15,000 different kinds of mice. Each one of them is genetically distinct. And we've been calling them the Jedi mice for genetic diversity. It turns out they're incredibly valuable because each one of them has a completely different response to all sorts of things. They have different personalities, they have different food preferences, they weigh different amounts, and they also tend to get different kinds of disease. So when I was up against this, I thought, okay, how are we going to quickly as we can, get a mouse that is susceptible to the SARS-CoV-2 virus, but has different host genetics, namely like people, different backgrounds. And so the way we decided to do this was to take our Jedi mice, and every one of those mice on the right, those are girl mice, was going to be mated to a boy mouse on the left that carried that transgene. So what that meant was that all the progeny of all those crosses were mice that had half of their genetic material coming from the mouse that we started with, the K18 mouse. And that included the transgenic part that allows the mouse to get infected. And every cell in every one of those mouse bodies has this transgene. But the difference is that the other half of the genome is variable. So it's really a lot like what happens when, let's say, someone gets divorced and they have children by another spouse. And the children all have the genes of the parent, but from, let's say, the father, but they may have different genes from the mother. So that's sort of the way to think about this. These animals are closely related to each other because half of their genome is identical, and yet the other half is not. I have to say that when I did this experiment, it was a quick and dirty thing to do. I thought it was easy. You just made a whole bunch of mice. They love doing that. But ultimately, the question was, are we going to see anything different? We still have half of that genome, of that gray genome, and we know that that mouse is really badly susceptible. Are we going to see anything that mitigates that severity by simply adding some more genes in from another mouse? And the answer was, you bet. I wouldn't be sitting here if it weren't true. But I have to say that this wasn't an easy experiment. The mice part was easy. The virus part was not. If you want to do an experiment with a virus like SARS-CoV-2, you do not do it at home. And by home, I mean my laboratory. You need to go to a very fancy containment facility where scientists have to dress up in these kinds of zoot suits with positive pressure so they look like Michelin men. And they work in extraordinarily contained environments to make sure that none of the pathogenic material gets out. And you need about 6 months training to go into one of these things. You can see how this guy is essentially like an astronaut, has his own respiratory setup. Now, fortunately, I had a friend who works at the Rocky Mountain Laboratories in Montana, which is run by the National Institute of Allergy and Infectious Diseases. And I called her up, Sonia, she's fantastic. And I said, Sonia, I need some help. Would you be willing to infect a whole bunch of mice that are all different with SARS-CoV-2 and see if they're any different? And she's a good risk taker. And she said, oh, well, why not? Let's just do it. So I sent her all the mice out to the Rocky Mountain labs, and she infected them along with her colleague, Shelley Robertson, with the SARS-CoV-2 virus and waited to see what happened. And about a month later, she called me up and said, you will not believe this. Every one of them is different. So we hit the jackpot. So I'll tell you how to read this little slide. The middle one that's sensitive is the good old K18 ACE2 mouse that I told you about on what's called a black 6 background. That's where it was made. So that's the gray background. Now look on the left. Now we have a mouse that's half of the gray mouse, and the other half is coming from a mouse called PWK. Doesn't matter. But look, one mouse died out of like 20. They all just kept going. That survival curve on the bottom tells us that the mouse is getting infected, because we knew that from other things we did, but it isn't getting sick. And then on the right, there's another strange mouse. In this case, we crossed the original mouse with NOD, another mouse strain. And look at that one. All the females were fine and the males died. So that was really strange. And these mice are not that far away from each other. Most of the mice we have are quite closely related to each other. And indeed, you don't have to read all of this, but just to give you the impression of just how different those survival curves are. Now, what you're looking at in each case is survival on the left and the weight loss on the right. You can see how weight loss tends to follow survival. For those mice that do survive, the few mice that do survive, for instance, in the sensitive strains, they do actually regain their weight. So you see a few of these lines coming back out on the right panels. But in general, I've sort of grouped these so you can see the resistant ones are in blue, the sensitive ones are in red, and the sex-biased ones are in orange. And here's the interesting thing. I showed you the NOD, that's the middle one. That's the one where the females do well, but the males do not. But look at the one on the left. On the left is cast, and the females do well, the males do even worse. And on the right, WSB, it's the females who are dying and not the males. So we really had a whole slew of models that we could then use. And this is just to give you a feel for it. If you just read the outcome, the symptoms were variable, and so was the outcome. So that meant that although we had the same virus in all the mice, with 8 genetic backgrounds that were different, we got 8 distinct disease profiles. So if that isn't a sort of smoking gun for genetics being important in this disease, I don't know what it is. Now, why? Of course, the next question is, how does this work? And here's a clue. Again, this is all unpublished data I'm showing you, so it's very raw, but I thought it would be interesting to see where we're at. It's such a fast-moving field that everything is happening sort of right away. So what we noticed was a particular protein that's extremely important in the immune response, called interferon alpha, was very, very high in the circulation of the mice that were resistant. And you can see that on the left. And it's just male in blue and female in pink. And you can see high levels in all of these animals. And then as you see the sensitive ones, they have much lower levels of interferon alpha. And then the sex-biased ones play out the way you'd expect. So is this likely to have any relevance? Well, a wonderful set of results from Akiko Iwasaki in Yale on looking at patients now showed that in the bloodstream of patients, those that did very well and rapidly cleared the virus over there on the left and had mild disease tended to have very high levels of interferon very early. And those interferon levels I showed you was only after 3 days in the mice, and the same thing is true in the human. Whereas on the right, you're seeing another kind of pattern. I mean, these are obviously idealized curves, but it gives you the impression of the data. There was a delay in the interferon response. It's like the immune system didn't wake up in time. And the virus just kept coming and coming and coming. And because there was nothing stopping it, it rose to very high levels. And then you end up with virus for a very long time because your immune system doesn't kick in fast enough. So you end up with inflammation and severe disease. Now, this isn't going to be all about one protein, but it's just one of the proteins that's very important in the response. And it's almost like a marker of immune response. And so the first lesson learned is how fast you respond to this virus is going to make a big difference. It's that early moment when your body says, oh my goodness, we've got a virus, and it kicks into gear. And what you see on the left is, well, it gets going, but by the time it gets going, the virus is really high. And the second piece of data that we were very excited about was how much this looked just like the mice. So this meant that we had models that we could use to study this disease and to study the variation in the disease. So now came the next phase, which was variants of concern. You probably all remember when the variants of concern showed up. We had alpha, we had beta, we had delta, we had gamma, we have Omicron, and now we have 50,000 kinds of Omicron. These variants are arising because viruses are extraordinarily fast to mutate to get around problems that they have with their host. So for instance, if the host isn't transmitting the virus fast enough to the next person, the virus mutates around until the virus that gets better at getting transmitted is then the dominant virus that ends up replicating and becoming the prevalent virus in a pandemic. And so these variants, as you know, have been coming in these waves, and then they get replaced by another variant that's even better. The good news is that in general in virology, the variants tend to end up being much more transmissible, but in general, less severe. And that's because viruses don't have any real reason to kill you. All that happens then is they don't have anybody to infect anymore. So the perfect host for a virus is someone that gets sick and gives it to 1,000 people, but doesn't necessarily succumb to the virus, but allows the virus to spread. And so here what we're seeing is an experiment that we did with our friends at the Trudeau Institute in New York with variants of concern, which they had been working on. And we asked a very simple question: can we possibly get around this business of humanizing the mouse and see if any of our mice are naturally susceptible to VOCs, to variants of concern? And to cut a long story short, we looked at beta and gamma, which were the prevalent ones when we did this experiment about 9 months ago. And what we discovered, to our surprise, was that several of those animals that I've just shown you turned out to be naturally susceptible to the viruses called beta and gamma. And you can read it now because you know how to read survival curves. So on the left, you're seeing the Castaneous curve, which is the most susceptible mouse to this virus. And by the way, that one was also susceptible when you looked at the virus infection of the hybrid mouse between the Castaneous and the K18. But you can see here, there's a survival curve of the original mouse as just comparison in the black. And you can see that basically what you've done is to find a way to make a mouse, or to find a mouse that was as susceptible to the virus, even without a human gene, as the transgenic mouse. Now that's great, because now we have a mouse that is naturally susceptible and therefore is probably going to be a better model. But on the other hand, I can tell you it cast a big spell over our labs, because if the Castaneous mouse can get it, then supposing someone wanders into the mouse room and they've just gotten a beta infection and they don't know it, all of our mice are going to get it and they're all going to die. So all of a sudden, we went into high gear with security. And on the right, you see another version where, again, the Castaneous is also, it seems to still be susceptible to the virus all on its own. And another mouse called PWK seems to have a little bit of susceptibility on its own as well, but not nearly as much. And all of those lines at the top of this graph are all the mice that you can see listed down on the right side of this slide that didn't have any susceptibility at all. They all got infected, but they didn't get sick. So now we have mice that do get sick, that don't get sick, that get sick on their own. What's going on? One thing that we noticed was that the literature was beginning to pick up on the fact that mouse ACE2 had sequences, remember those letters that I showed you at the beginning, that apparently were recognized, especially by the Omicron variant. And so maybe what was going on was the virus was mutating its spike protein to make itself more transmissible, but in the process, just by happenstance, managed to change itself so that now it could recognize mouse ACE2 and infect mice. And what we've seen recently in our work is that these viruses of concern, variants of concern, they infect different tissues in mice like the cast, which are susceptible. So you see over on the right, that's a blood vessel in the lung. And you can see how the epithelial cell is really cranking out the virus in purple. Whereas the original mouse that I described to you, the one that dies so fast, doesn't seem to have that same pattern. The blood vessel isn't really where the virus is sitting. So there's tons to be learned. We now know that we have to look at where the virus goes, how fast it goes there, and what does it do when it gets there. And then we might really understand what's different about people that get very sick versus people that don't. So now what we basically have is a situation where we have mice that we can pull out of the shelf and say, this is a mouse that won't get sick with your virus. This is a mouse that will get sick with your virus. And you can study the differences. And so obviously this is a real boon to the community to be able to work with this wonderful, diverse collection of animals. And of course, this is a real picture. The mouse was really sitting there like this. It's a little WSB mouse. He always has a little star on his head. And so the concept is we might have in our collection of 15,000 mice one mouse for every virus of concern or variant of concern. And that's the hope we have of actually being able to make a much better and more rapid progress on the next pandemic. Furthermore, I just showed you 8 mice, but we have 15,000, and we can make even more genetically diverse ones and test them and hopefully find new mice for every pathogen and for accelerating drug discovery, because obviously it's much faster to give mice the drug than humans. Ultimately, we'd love to know more about long COVID. So we've initiated a study on long COVID to see if we can pick up symptoms in mice that survive the initial infection, but then might end up with a lung infection or some other problem, a fever or nausea. And we have ways of testing mice for those kinds of readouts. And with that, we might be able to crack the long COVID mystery. Ultimately, we're not going to get away from viruses. They're here to stay. Here's a dizzying and somewhat depressing list of all the viruses that we end up with today. And many of us have had all these viruses. Hopefully, some of us will never have polio or some of the other ones, but these are a rapidly evolving picture of the viruses that plague us. But I'd like to just end with one interesting fact, which is that getting rid of all viruses would actually not be the best thing for the human race. And that's because many of the adaptations that we have made as a race, as we evolved away from the primates and ended up migrating out of Africa and all over the world, a lot of what changed in our various ways to adapt to our new environments came about through viral infections that challenged us to actually have more robust immune systems or more robust ways of fighting off certain kinds of pathogens. Furthermore, viruses move genes around between people. They dump their material into your genome. And a lot of what we've got in our genome is old viral DNA that we've actually repurposed. So they've actually contributed to human evolution in a remarkable way. However, what we really want are the viruses not to do anything to us that makes us really sick. And for that, outbreak science is obviously something that is really now, I think, on the agenda for most research institutes. And the theory that we have at the Jackson Laboratory is if we use mice to model humans in a way that will allow us to go backwards and forwards, testing our hypothesis from the humans in the mice, and then looking to see if what we see in the mice is true in the humans, I think we can get to cures a lot faster. At least that's the hope. And this is just a picture because I really love the concept of having a mouse for every person and making models as unique as we are. And here is another group of unique people. In this case, they're people, but we call ourselves the COVID Mousers. Those are all people at the Jackson Laboratory in the top. I don't have time to tell you about each one of them, but there is amazing talent among this group. I'm also very grateful to the Rocky Mountain Laboratories and the National Institute on Allergy and Infectious Diseases. Steve Holland is the chief and has been very generous funding much of the work you've just seen. And of course, I mentioned Sonia and her colleague, Shelley Robertson. I've also had fantastic advice from a number of my colleagues at Imperial College and in Cambridge. And I would also like to mention the fact that more recently we've started working with Trudeau colleagues, Bill Riley and Atsuo Kuki. So finally, just like to thank the mice for being so amazing and different. And thank you for listening. And it's just a picture of the Jackson Lab. Do come visit us. It's beautiful up here. Thanks a lot.